Hi friends, today we are discuss about the topic ultrastructure of viruses. We know that viruses is an infectious agent that replicates only inside the living cells of an organism. And we first talk about the search for the elusive virus. First, in 1884, Louis Pasteur postulated that rabies was caused by a virus, and 1819, Ivanovsky and Beijing showed a disease in tobacco was caused by a virus, and virus is a non-cellular particles with a definite size, shape, and chemical composition. A foundation of virology. Virology means study of viruses. Non-living agents that infect all life forms. Example: phages versus animal viruses. And these animal viruses go on attack animals, while the phages is an attack bacteria, so that be called bacteriophage. Approximately 1,500 non-viruses and estimate 4 lakh exist. And visualization of viruses is used for electron microscopy. And electron microscopy is used to gather much more information of viruses and different body components in case bacteriophage. And this is a typical structure of a bacteriophage. It's have head, collar, core, and helical sheath, and tail pins. And general characteristics of viruses. And viruses have different size and shapes. This is a round shaped virus, and this is a round shape, and this is a coil type virus. And each virus is obligatory intracellular parasites, and it is filterable. And in Latin, viruses is a Latin word. Virus means poison. It contains DNA or RNA, and not in both. And it's contain a protein coat is called capsid. Capsid is made up of capsomeres in various shapes, and some are enclosed by an envelope. Envelope is the outermost covering of viruses. The viruses that contain envelopes are called envelope viruses. The viruses in which envelope is absent is called non-envelope viruses. And the envelope viruses is made up of lipoprotein molecules. And some viruses have spikes, and most viruses are tissue specific. Means that means some certain viruses will infect with a certain type of tissues. Example: hepatitis infects the liver tissues. Viruses shapes and sizes. There are different shapes of viruses. Most animal viruses are spherical in shape. And different shapes of viruses and their examples um, are first tobacco mosaic virus. Tobacco mosaic virus shape is a round shaped virus. It's a round shaped virus. This is tobacco mosaic virus. And pox virus is a brick shaped virus. Pox virus is a brick shaped virus. And rabies virus is a bullet shaped virus. And Ebola virus is filamentous in shape. And coronavirus petal shape. And rotavirus is a wheel shaped virus. And And next is size of viruses. Viruses are we know that viruses are smaller than bacteria. They are filterable, and size of viruses is measured by using ultrafiltration, ultracentrifugation, electron microscopy. And the size of virus is a range from 20 nanometer to 300 nanometer. In size of parvovirus is at 20 nanometer, and it is the smallest virus. And the size of pox virus is 300 nanometer, and it is the largest virus. And viruses are polyhedral. um, why it is polyhedral? Because of the different planes they have. They can be either round shape missile virus, and isohedral viruses are poliovirus and parvovirus. Polio and parvovirus are isohedral virus without the capsule, but so they are naked viruses. And helical viruses is tobacco mosaic virus, rabies virus, and measles virus. And isohedral viruses is uh, poliovirus, parvovirus, and herpes simplex viruses, and complex virus. And complex viruses which does not have any fixed geometrical structure. They have multiple structure linked viruses. Example, bacteriophage. The head region of the bacteriophage is like isohedral viruses, and the collar region is like helical viruses. So why we called the bacteriophages is complex virus. Viral structure. The structure is divided into two types, covering and central core. And covering is divided into capsid. We know capsid is made up of capsomeres and envelope. Envelope is not found in all viruses. Envelope is the outermost covering of viruses, and the virus that contains envelope is called envelope viruses, and envelope is absent virus is called non-envelope viruses. And the central core consists of nucleic acid molecules, these are DNA or RNA, and matrix proteins, enzymes, and they are not found in all viruses. And we know that viruses have now replicate herself, and they have no rules, and they can only make copies when they attack their host. And once they attach with their host, they hijack the protein synthesis machinery of their host, and then uh, then the viruses utilizes the host enzyme for the protein synthesis. General structure of viruses is capsids. Capsids is made up of protein called capsomeres, and all viruses have capsids. The capsids together with the nucleic acid is nucleocapsid. Some viruses have an external covering called envelope, and those viruses have doesn't have envelope. They are naked, and each capsid is made up of capsomeres. And two structural capsid types. One is isohedral symmetry and helical symmetry. It is made up of 12 vertices and 20 sides. Isohedral symmetry is made up of 12 vertices and 20 sides, and it shows two types of capsomeres. Structure of viruses. We also say that The protective shell is called capsid. As capsid is made up of many identical protein subunits, and the capsid is protect the genetic material, and capsid is symmetrically organized, isohedral and helical, and the weight is 50%, and enveloped or non-enveloped. Envelope means is a lipid containing membrane that surrounds some virus particles. The envelope is located outside the capsid, and the next is function of capsid or envelope. Protects the function of capsid or envelope is protect the nucleic acid when the virus is outside of the host cell, and help the virus bind to a cell surface and assist the penetration of viral DNA or RNA into a suitable host cell. That is a function of capsid and envelope that protect the virus, nucleic acid of viruses. And general structure of viruses. Nucleic acids. Viral cell have only one type of genetic material, and it may contain either DNA or RNA, never both. And virus does not contain the enzyme that are important for replication and multiplication of viruses. So virus cells are dependent on host cell for its multiplication and replication. And difference between DNA viruses and RNA virus. DNA virus is usually double stranded. We know that usually double stranded, but maybe single stranded and circular or linear. RNA viruses is usually single stranded, maybe double stranded, maybe segmented into separate RNA pieces. And RNA genomes ready for immediate translation are positive sense RNA, and RNA genomes that must be converted into proper form are negative sense RNA. And all DNA viruses are double stranded. We said that all DNA viruses are double stranded, and except parvovirus. In exception case, parvovirus has a single stranded DNA. Host range and specificity is the another very important properties of a virus. Virus has its specific host range, and it can be narrow or it can be broad. You can see this picture. Is generally virus host range is narrow than the bacteria, because they can only attack a particular host. They can only attack a particular host, and this specificity is known as tissue tropism.